We were studying plant kingdom. Today we will be discussing about bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks, soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect, and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophytes is haploid. It produces gametes and is called gametophytes. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellated antherozoids. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoids are released into water where they come in contact with archegonium. An antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free-living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division, meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of Sphagnum, a moss, provide peat that have long been used as fuel, and because of their capacity to hold water, as packaging material for transshipment of living material. Mosses, along with lichens, are the first organism to colonize rock and hence are of great ecological importance. They decompose rocks, making the substance suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid. Example, Mercantia. The thallus is dorsiventrally and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structure called gammae, singular gemma. Gemmae are green, multicellular asexual buds which develop in small receptacles called gemma cups located on the thalli. The gemmae become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Mosses. The predominant stages of the life cycle of the moss is the gametophyte, which consists of two stages. The first stage is protonema stage, which develops directly from a spore. It is a creeping green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organ antheridia and archegonia are produced at the apex of the leaf shoots. After fertilization, the zygote develops into a sporophyte, consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Now we will discuss about pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamental. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in the next chapter. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. The leaves in the pteridophyta are small, microphyllous, as in Selaginella, or large, macrophyllous, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophyles. In some cases, sporophylls may form distinct compact structure called strobili or cones, Salginella and equisetum. The sporangia produce spores by meiosis in spore mother cell. The spores germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow because of the specific restricted requirement and the need for water for fertilization. The spread of living pteridophytes is limited and restricted to narrow geographical region. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of anthrozoids. The male gamete released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium results in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro, large, and micro, small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes, respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit. considered an important step in evolution. The pteridophytes are further classified into four classes: Coelopsida or Coelotum, Lycopsida, Selaginella and Leucopodium, Sphenopsida, Equisetum, and Pteropsida, Bryopteris, Pteris, and Edentum. Gymnosperms. The gymnosperms. Gymnus means naked, sperma means seed. These are the plants in which the ovules are not enclosed by any ovary, wall, and remain exposed, both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree, Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, for example Pinus, while in some other, like Cycas, small specialized roots called coralloid roots are associated with enterofixing cyanobacteria. The stem are unbranched in Cycas or branched in Pinus and Siderus. The leaves may be simple or compound. In Cycas, the pinnated leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. The thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid macrospores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains take place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree. In Pinus, however, in Cycas, male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus, nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovule are borne on megasporophylls, which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspore enclosed within the megasporangium, nucellus, develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and the female gametophyte do not have an independent free-living existence. They remain within the sporangia, retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovule and discharges their content near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and ovule into seeds. These seeds are not covered.